All right, thank you. So my name is Matt Britz. I am the President and Chief Operating Officer of AffyImmune, and I'd like to take a moment and thank the organizers for inviting us back to speak. Since last year, we've made some really good clinical progress, and I am excited to share that today. So a quick overview about AffyImmune. One of the things that we are looking to do is expand and extend the successes that we've seen in the CAR T space in hematological tumors to the solid tumor space. And right now we have a platform that we use to address these challenges. It's called Tune and Track. I'll go into great detail about that later, but basically we affinity tune the interaction between CAR T cells and the cancer antigen. And we also have, we co-express membrane protein on our CAR T cells that allows for real-time tracking in the human body. We are currently in the clinic. We have a lead asset that we call AIC-100. It is an autologous CAR T cell therapy that targets ICAM-1. It is currently enrolling, and we currently have 4 U.S. sites here in the U.S., and we have Fast Track and Orphan Drug designation. And what we debuted at ASCO here in a quick summary in the summer is that we had a partial response in an anaplastic thyroid cancer patient at an intermediate dose level. So we are currently raising a Series B, so I need to tell everybody that we have a first-in-class, best-in-class opportunity. Specifically in anaplastic thyroid cancer for wild type, there is no current standard of care. So there is potential here for an autologous CAR T to be first line. In poorly differentiated thyroid cancers, there is an approved therapy. However, upon relapse, these patients would be CAR T eligible. With the phase 1 trial that's going on right now, we have a lot of key data that will be coming out over the next several months, and we have a unique platform that's called Tune and Track, although I think from the last talk I should be calling it a scalable product engine, I believe was the term, so I will try to incorporate that going forward. In terms of fundraising, our last round was a $30 million Series A that was funded in October of 2021. And that was led by our majority shareholder, Ori Capital. All right, so in terms of solid tumor CAR T, it's pretty well known what a lot of the issues that have cropped up in solid tumor CAR T are relative to the liquid tumors. And the number one challenge for these CAR T cell therapies in solid tumors is the on-target off-tumor toxicity. When you have something like CD19, which is with the target of blood cancers, or BCMA, you can wipe out all of the B cells in the body. There's no expression of these targets on any other tissues, and when you wipe out all of the B cells, the patient survives. Unfortunately, in solid tumors, we have not found that magic target that is only expressed on cancer cells but not expressed on normal tissues. And so if you don't have your affinity, if you don't have your CAR T cells affinity tuned correctly, what happens is the CAR T cells will go in the body and they'll kill the cancer, and that's great, but they'll also identify the antigen on normal cells and they'll kill those as well. And that's resulted in significant toxicities. You've probably heard of CRS and ICANS, and we have seen some patient deaths in solid tumor trials for that reason. All of these other issues are significant for solid tumor companies, and I won't go into detail about every single one. I will say that antigen loss and tumor heterogeneity, target selection plays a really important role in limiting that. T-cell exhaustion, affinity tuning really helps with that as well. And for all of these, it is our goal at AffyImmune to have something in the pipeline that addresses all of these key challenges. So this is introducing our Tune and Track platform. The Tune stands for affinity tuning, and all that really is when creating a CAR T cell binder for the cancer antigen, what we do is we have a process by which we tune that affinity low enough that it still kills the cancer cells, but high enough that it still kills the cancer cells, but low enough that it does not recognize the cancer antigen on normal tissues. And whenever you affinity tune, not only do you get that better separation of killing, but it also results in better, less exhaustion and better serial killing. The tracking part, and this is really cool because I'm convinced we're the only company that's doing this, is we express SSTR2 on our CAR T cells. It's a membrane protein and it interacts with gallium dotatate. Gallium dotatate is an FDA-approved imaging agent, so it's already approved, and what you can do then is during your trial, you can do a standard FDG PET that shows you where the tumors are, and then you can do a dotatate scan, and that'll show you where in the body the CAR T cells are. And when we originally started out, we thought that this might be a good safety protection. You could do a dotatate scan, you could see the CAR T cells. If they're collecting somewhere where there's no tumor, you can then alert the PI, hey, CAR T cells are in the lung, you might want to worry about lung toxicities. But in the patients that we've dosed, we've actually never seen this. We've seen perfect alignment between the CAR T cells and the tumors. And this is basically how we go about affinity tuning. This is a good chance for me to mention one of the other things that we do that's a little different than everybody else in the field is a lot of CAR T companies will use an antibody fragment to drive their CAR T cells to the cancer cells. As I mentioned, we're targeting ICAM1, and ICAM1's binding partner is a molecule called LFA1. And so our CAR T cells actually aren't antibody-based. We use an affinity-tuned version of the I domain of LFA1 to engage the target. And if you take wild-type LFA1 and you arm a CAR with it, it's a very low affinity, and what happens is the tumors just grow out of control. You basically get no efficacy because the affinity's too low. If you do what I said about what most people do, and that is get a very high-affinity antibody, the high-affinity antibody causes toxicities, and rather than having tumor killing, you have toxicity and mouse death anyway. So that is not ideal. And then we had another variant here that we call F292G. This is a variant of LFA1 as well. I call it medium to high affinity. And what happens in vivo is you get good initial response, but what happens is the affinity is still too high. You get exhaustion, and that allows the tumors to come back. And F292A is a variant that ultimately became AIC100, which is our product in the clinic now. And when you get it just right, you get good killing throughout and no toxicities. Another part of that slide I forgot to mention was that ICAM1 between mouse and human is reactive, so it actually is a good toxicity model. In some cases, that's not always true, and so that's not necessarily a good toxicity model for everyone. All right, ICAM1, another reason that we picked it is one of the things that you need to do in solid tumors is you need to ensure that you have differential expression between your cancer cells and your normal tissues. And you can see here that in tumor mets and malignant tumor, you have very high expression levels compared to normal cells in thyroid cancer. Another reason is that ICAM1 is associated with poor prognosis. Going back to the CD19 example, one of the things that you see in CD19 cancers is that after CAR T cell therapy that you kill all the cancer cells that express CD19, but since CD19 isn't actually, it's just a marker, it doesn't do anything that causes cancer, often you have CD19 negative relapse in these patients. And so when you're looking at solid tumor targets, one of the things we like to look at was we're looking for something that is associated with poor prognosis, that is associated with metastasis. So that way, when you do kill off all of these ICAM1 high cells, the tumor has fewer ways to be able to evade antigen escape or to create antigen escape. Another thing about ICAM1, and I think this is unique to ICAM1, I don't know of any other targets that do this, but what happens is upon CAR T cell engagement with a cancer cell, interferon gamma is released, and the cancer cells, in a way to avoid killing, start downregulating their interferon gamma receptors. But what happens in cells whenever you downregulate interferon gamma is you actually increase the expression of ICAM1. And so upon tumor engagement, we actually get forces that begin to work to encourage the cancer cell killing. And so I think that's unique to ICAM. In terms of pipeline, so really we are focused very strongly on getting our lead product AIC-100 to approval in thyroid cancers. You may know thyroid cancers is very small. It's a very small market, and there are other cancers that express ICAM1, 60, 70, 80% of other cancers with high ICAM1 expression. So we are currently undergoing an indication expansion for undisclosed indications. And that's our next major effort, and we expect to have IND filings in the middle of next year. And then because it is a platform, we can tune and track basically any target with differential expression, and we have a research program in an undisclosed target and an undisclosed indication. So as I mentioned, AIC-100 is in the clinic. This is a schematic of the phase 1 study design. It is a modified 3+3 dose escalation study and limited currently to anaplastic and poorly differentiated thyroid cancers. One of the things we are doing a little differently is we do not dose based on weight or body area. We do flat dosing. It's just easier from a manufacturing standpoint to make bags with 10 million, 100 million, or 500 million CAR-T positive cells. And like any phase 1, the objective, the primary goal is safety, but we are looking for early peaks of efficacy using RECIST 1.1. And we scan week 2, week 6, and every 2 months for a year. We expect to finish the phase 1 data in the middle of next year. So this is what we released at ASCO from our poster, is that we have had good safety thus far. We have not had any DLTs. We've only had mild CRS that resolves on its own. We also have, all 3 patients at dose level 2 had tumor shrinkage, and 1 patient had a partial response with a 42% reduction in the tumor. This was also confirmed at month 3. So to show you a little bit about how FDG PET and the dotatate scans work, if you look here, I don't have a pointer, but if you look here on the left, so if you're unfamiliar, SUV is just a unitless measure of glucose uptake or glucose use. You can see at the baseline, we have a lesion there in the lung. And then at week 2, we actually have an increase in glucose utilization in the lung. And this is called pseudoprogression. This happens in solid tumor trials all the time. And the reason for that is not only do cancer cells use more glucose than those cells around them, activated T cells also use more glucose than those cells. So it looks like that your tumor has actually increased at week 2, but that's pseudoprogression. It's due to the tumor, the CAR T cell infiltrate. And then you can see at week 6, we now have SUV down to 3.1. So we have significant tumor reduction, and it's measurable not just in size, but also in glucose uptake. On the right side there, you can see the dotatate scan. This is a little different in that people naturally express SSTR2 at varying levels, so you absolutely have to have a baseline, and it's different for pretty much every patient. And so you need to get a baseline. You can see at week 2, we can see CAR T cells. Now we're just measuring only CAR T cells. We have CAR T cell infiltration into the tumor, and then at week 6, we have almost back to baseline, still above baseline. That's a combination of both fewer T cells there and also the tumor reduction. This is a summary of the clinical progress thus far, released at ASCO this year. No safety, no DLT issues. At dose level 1, which is a very low dose, we had 2 progressive diseases and a PI-determined stable disease. But at dose level 2, we had 3 patients, all with tumor reduction, and then we had the partial response in the anaplastic thyroid cancer patient, 42% tumor reduction at day 42, confirmed at month 3. So I mentioned earlier that ICAM1 was expressed in multiple other kinds of cancers, and I have cleverly disguised them here as indication 2 and indication 3. We have not disclosed them yet, but these are cancers that have larger markets and significant unmet need as well, and we plan to enter these, IND these next year and begin trials either late in 2024 or early in 2025. it's a land and expand strategy, we're calling it, to go in a smaller indication and to expand it into larger indications. So I mentioned first in class, best in class. What we want to do is become one of, if not the first CAR T company to be approved in solid tumors. And you may say there are people that are way far ahead of you, and while that is true, we believe that by choosing a smaller indication, we will be able to get approval on a lower number of patients initially. And of course, as I mentioned earlier, going forward, we have very good data and news flow coming out here in the next several quarters, and we have a great platform. And I am out of time. So thank you for your time and attention. And if you have any questions about AffyImmune, please find me at the events. Thank you.